So, dear students, good evening. So, today I will discuss transcription in prokaryotes. So, what is the transcription? So, you see the present slides. So, this is the central dogma, and already I was telling you. So, DNA after replication will give rise to the DNA. So, from DNA to DNA, that is called replication, and from again DNA to mRNA, that process is called transcription. And when mRNA will translated, it will give rise to the protein. So three processes there, that is called replication, transcription and translation. And maybe sometimes mRNA also transcribed to the DNA, that is called reverse transcription. So today I will see, or you will see, what is the transcription. So I will discuss basic principle of transcription. So transcription is the nothing but synthesis of RNA molecules using DNA strands as the templates, so that the genetic information can be transferred from DNA to RNA. So just I told you, whatever genetic information lying in the DNA, that will be transcribed to the RNA, or that will be transferred to the RNA. So the main question is that, so what is the similarity between replication and transcription? So if you see, so both processes, that is called replication and transcription, use DNA as the template. Okay, so in both cases DNA acts as a template, and in both cases there is formation of phosphodiester bonds. And also third similarity is the direction, that is called synthesis of transcription or replication, always takes place from 5 prime to 3 prime. So these are the similarity between replication and transcription. So sometimes you may be asked, what is the similarity? So this is the now you can see the differences. So this is the replication, and next slide is transcription. So if you see basic differences between replication and transcription, so in terms of template, in case of DNA replication, always you are using double stranded. So DNA is always double stranded. But in case of transcription, your DNA should be single stranded. Okay. But no doubt, here also melting will take place. But here DNA will be double stranded, and here your DNA will be, template will be not DNA, template will be single stranded. Now substrate. So in case of replication, that is called deoxynucleotide triphosphate, but here only NTP, because here we are going to form RNA, that's why only NTP, not deoxynucleotide triphosphate. And primer. So in case of DNA replication, that is called RNA primase is forming or forms primer, that is called RNA primer. So here primer is present, but in case of transcription there is no primer, there is no formation of any primer. If you see the enzyme, so in case of replication, the DNA polymerase is important enzyme, but in case of transcription, RNA polymerase is important enzyme. Although we will discuss in case of prokaryotes and eukaryotes. So in case of prokaryotes there is single RNA polymerase. In case of eukaryotes, again there are different types of RNA polymerase, like RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. So we will discuss again later on. Now if you see the product, so in case of replication, the product is double stranded DNA. So this is the basic difference or main difference. So here after replication, again you are getting the double stranded DNA, but in case of transcription you are getting single stranded RNA, because again for translation you need to have single stranded RNA. And again base pairing. So I was telling you, in case of base pairing, here important is that adenine pairs with thymine, but in case of transcription, adenine paired with uracil. So these are the fundamental differences between the replication and transcription. So sometimes you may be asked, write the differences between the replication and transcription. Now, the whole genome of DNA needs to be replicated. That is again difference, because whenever DNA replication takes place, whole genome of DNA is replicated. Like you might have studied the cell division. So G1, S, G2. So the S is called synthesis phase. But in here, only a small portion of genome is transcribed in response to development requirement, physiological need, and environmental changes. So maybe some genes are transcribed, not always genes, not all genes are transcribed at the same time. Because based on the requirement, based on the need, like there is housekeeping genes. This is always transcribing. That is always constantly expressing. That is called housekeeping genes. So that is very, very again very, very important difference. Now the DNA regions then can be transcribed into RNA or structural genes. So again, uh, these are also important terminology. Like in case of operon, structural genes are transcribed together. So now we will discuss what is the template. So template strand is the strand from which the RNA is actually transcribed. Okay, so when RNA is actually transcribed on the strand, that is called template strand. And it is also known as antisense strand. But what is the coding strand? So coding strand is a strand whose base sequence specifies the amino acid sequence of the encoded protein, and therefore it is also known as sense strand. See the diagram. If you see the diagram, so this is called coding strand, and where that region will transcribe, that is called template strand. So after transcribe, you can see. So sometimes questions is asking. So this is the DNA strand. So what will be the RNA nucleotides? Okay, so you can see the difference. So here C is G, C pairs with G, and again G with C, then again T with A. So students often confuse. They are putting T with U like that sometimes. So T always pairs with A, but difference is that you can see here. So T pairs with U. Here you can A pair with U. Generally A pairs with T, but in case of RNA, A pairs with U. So remaining all as you can see. Where A is there, it pairs with U. A U, A U. So this is the basic difference, because sometimes in the examination or net examination, like suppose you will be given or you are given DNA strands, so you have to write the RNA strands. So the strands often confuse. So be careful. Now we will discuss what is the asymmetric transcription. So only the template strand is used for the transcription, but coding strand is not. Both strand can be used as the template. Yeah, it is possible. Transcription direction on the different strand is opposite. Okay, suppose when two strand is there, sometimes one such transcription is taking place in one direction. So in other strand, transcription taking place in another direction. That is called opposite direction. So this feature is referred as asymmetric direction. Asymmetric direction means one from this side, another from that side. Means both are going opposite direction. That is called asymmetric transcription. So just you can see the example, like you can see. So this is the coding strand, this is the template strand. Again, this is the template strand, this is the coding strand. So like you can see, this is the template strand. So this is here transcription will take place in this direction, and that is the template strand. So transcription will take place in that direction. So that is called asymmetric transcription. So this is the example. Like in case of adenovirus, you can see ki at the above side transcription is you are having transcription transcript in one direction, and in the other template strand transcript are in different direction. So now DNA polymerase, but here you are not going to use DNA polymerase. Here you have to use RNA polymerase. So sorry. So enzyme responsible for RNA synthesis is called DNA dependent RNA polymerase. So this is called DNA dependent because it is dependent on the DNA. So DNA dependent RNA polymerase. And again, prokaryotic RNA polymerase is a multiple subunit protein. So it is around consists of 480 kilodalton. Okay. So again here I am telling difference in case of prokaryotes. Only RNA polymerase is involved. But in case of eukaryotic system, we have three different types of RNA polymerase. So this is the basic difference I was telling you. So they have three kinds of RNA polymerase, and each of which is a multiple subunit protein and responsible for transcription of different RNA, because you have mRNA, you have tRNA, and you have RNA. So for every RNA you have to have three different RNA polymerase. Okay, so now we'll discuss RNA polymerase in case of E. coli. So if you see, the holoenzyme of RNA polymerase in E. coli consists of 5 different subunits. So alpha, beta, beta prime, omega, and sigma. Okay, so these are the different units. So sometimes this question also may be asked in the examination. So you can see here, so how arrangements are there, like alpha to 2 alpha is there, that is called alpha 2, then beta prime, then sigma, and omega and sigma. Okay, so this is called omega. And if you see the different subunits and molecular weight and function, so all the subunits have different molecular function. Like suppose alpha, so it determines the DNA to be transcribed. Beta, it catalyzes the polymerization. Beta prime, it binds and opens the DNA template. Sigma, that is called recognizes the promoter for synthesis initiation. So what is the function of sigma? So just it will scan the promoter, and where it finds, then RNA polymerase will come and it just go out. That is called sigma. So one chemical, that is called rifampicin. It is the drug for the tuberculosis treatment and can bind specifically to the beta subunit of RNA polymerase and inhibit the RNA synthesis, because you have to stop the multiplication of the bacteria. So that's why rifampicin is used to control the tuberculosis disease. And RNA polymerase of other prokaryotic system is similar to that of E. coli, because I was telling you, or I am telling you, this is we are discussing transcription in case of prokaryotes. So we have different types of prokaryotes. So whatever function, whatever process involving in the E. coli, same process is applied to the other prokaryotic system. So now RNA polymerase of eukaryotes. So I was telling you there are different types of polymerase 1, 2, 3. So if you see the products, so in case of RNA polymerase 1, that will form 45 base RNA. Again, RNA polymerase 2, it forms heteronuclear RNA, it is called hnRNA, and polymerase 3 will form 5 base RNA, tRNA, and snRNA. So this is the very important sometimes, but if you see the basic, so RNA polymerase 1 forms rRNA, RNA polymerase 2 forms mRNA, and RNA polymerase 3 forms tRNA. So these are students are generally remembering these things, but 5s RNA and snRNA is also found by RNA polymerase 3. So that is again you have to keep in the mind. Now if you see the sensitivity, so one of the antibiotic, or that is called amanitin, this particular antibiotic is extracted from the mushroom. So based on the sensitivity, if you see, the RNA polymerase 2 is highly sensitive to the amanitin, and 3 is moderately sensitive to amanitin, and first is there is no sensitivity. So this is the difference according to the sensitivity to amanitin. See the diagram. If you see the diagram, so this is called coding strand, and here that. So now we will discuss prokaryotic promoter. So in case of prokaryotic promoter, you see the composition. So again I will discuss two words. One is called upstream and second is called downstream. So generally transcription starts at +1 site. It is called transcription start site. So if you go opposite direction, okay, like -1, -2, -20, that will called as upstream sequences or upstream portion. And if you go opposite direction, like +1, +2, +3, like that, so that is called downstream sequences or downstream portion. Okay, so here in case of prokaryotic promoter, you will find two region. That is called -10 region, and second is called -35 region. So -10 region is generally called as Tata box, because you can see TATAAT means it looks like Tata. That's why you can call Tata box. But this Tata box is what called discovered by Pribnow, one of the scientists, and that's why it is also known as Pribnow box. Okay, so these are the consensus sequences. So there are two words: conserved sequences and consensus sequences. So in case of conserved sequence, there is no change in the any what nucleotide sequence or even protein sequence. But in case of consensus sequence, means maybe they change in the nucleotide, but they have same function. Okay, so if you see the next slides, you can see. So it is written consensus sequences. You can see here. So this is the difference. So here we have compared with different types of promoter. Like you can see, -10. So we have -10 from the different promoters, like we have tree promoter, leg promoter, erect promoter, rhabdosis promoter. So if you see the Tata box, so here in case of you have TATAT here, okay, then TTAACT, then TATGTT, then TATAT, then TACDGT. So all the -10 region in every case like tree, leg, erect, they act as a Tata box. Although there is minor change in the sequence, but they act as a Tata box. So that is called consensus sequences. Okay, so sometimes question may be asked, what is the difference between conserved sequence and what is the consensus sequence? So you have to give the example. So conserved sequence means there is no change in the nucleotides. Okay, that's why it is conserved. Like you are telling that evolutionary significance, there are some domain motive. So just like motive. Okay, that is your conserved sequences. So that is the, you see the alignment of different types of promoter. So minor change in the Tata box. And again I am telling -35. So again here you will find consensus sequences. So you have TTGACA. You can see here GTGTCA, then TTGACA, triple TACA, then TTGATA, then CTGACG. So both the region that is called -10 and -35 both act as a consensus sequences. Okay, so now I think hope you are getting the points. Now -35 region of TTGACA sequence is the recognition sites and binding site of RNA polymerase. So this is the site for the recognition. And -10 region of Tata is the region at which a stable complex of DNA and RNA polymerase is formed. So this is called the region where a stable complex of DNA and RNA formed. Now we will see the process of transcription. So there are some general concepts. So transcription process takes place in 3 different phases. That is called initiation, elongation, and termination. Okay. So the prokaryotic RNA polymerase can bind to the DNA template directly in the transcription process. But in case of eukaryotic RNA polymerase, it requires various cofactors to bind the DNA template together in the transcription factor. We have different YouTube on the eukaryotic transcription. Okay, so you will see in detail in different YouTube. Okay, so this is the difference. So here prokaryotic RNA polymerase can directly bind to the DNA template, but in case of eukaryotic system, you have to have different cofactors. So one chemical that is called rifampicin. It is the drug for the tuberculosis treatment and can bind specifically to the beta subunit of RNA polymerase and inhibit the RNA synthesis. Because you have to stop the multiplication of the bacteria. So that's why rifampicin is used to control the tuberculosis disease. And RNA polymerase of other prokaryotic system is similar to that of or takes place. Now you see in detail how initiation is going to take place. So in case of initiation, you can see RNA polymerase recognizes the TTGACA. So just I told you, and slice to the Tata box, then opens the DNA duplex because your DNA is double stranded. So that is called melting. So on the, you can see here unowned DNA. So 17 base pair opened. That is called melting. So this is the DNA double helix. So unowned region is around 17 plus minus 1 base pair. So sometimes 16, sometimes 18. So it may be chances. So that is called initiation. Now what happens? The first nucleotide on RNA transcript is always purine triphosphate. That is also very, very important. So GTP is more often than ATP. And structure remains on the RNA transcript until the RNA synthesis is completed. So 3 molecules forms a transcription initiation complex. Like you can see here, 3 molecules: RNA polymerase, then DNA, then your RNA. So just I was telling you, so no primer is needed for the RNA synthesis. So sigma subunit falls from the RNA polymerase once the first 3' 5' phosphodiester bond is formed. So just means it just after initiation of transcription, your sigma subunit falls. Then coenzyme moves. So other factors and your RNA polymerase, it is called coenzyme, moves along the DNA template to enter the elongation phase. So there is just so now what happens in elongation phase? The release of sigma subunit causes the conformation change of the coenzyme, and coenzyme slides on the DNA template toward the 3' end. So free entropes are added sequentially to 3' OH of the nascent RNA. That's why I am telling you, always you have 3' OH, so that the incoming nucleotides will make the phosphodiester bond, and finally you will get elongated RNA strand. And RNA polymerase, that is called nascent segment of 14 nucleotides, and nascent RNA forms a complex called transcription bubble. So you have RNA polymerase, you have DNA segment, and you have nascent RNA. So all three will form transcription bubble. The 3' segment of nascent RNA hybridizes with the DNA template, and 5' end extend out the transcription bubble as the synthesis, and that is called processing. So you can see the transcription bubble. You can see you have your DNA strand, then RNA polymerase, then your nascent RNA. Okay, so this is the how looks like. This is the other card. So now we will discuss what is the template. So template strand is the strand from which the RNA is actually transcribed. Okay, so when RNA is actually transcribed on the strand, that is called template strand, and it is also known as antisense strand. But what is the coding strand? So coding complex. You can see so how transcription is going to take place. Then promoter clearance means here your sigma factor is going to be removed. Then transcription elongation place. So if you see the difference between transcription and translation in case of prokaryotes and eukaryotes, so just I wanted to tell you. So in case of prokaryote, transcription and translation always take place simultaneously. But in case of eukaryotes, first transcription will take place followed by translation. So again, this is the basic difference. So if you see this is the TEM diagram, that is called transmission electron microscopy diagram. You can see here. So how transcription and translation is going together. So this is the, and you can see here, this is the polysomes like ribosomes on the mRNA. So many mRNA is forming, it is called polysomes. Now termination is because after initiation and elongation, again transcription should be terminated. So RNA polymerase stops moving on the DNA template, and the RNA transcript falls off from the transcription complex. So termination may be done by either row dependent or row independent. So now RNA polymerase of eukaryotes. So I was telling you okay, there are different types of polymerase 1, 2, 3. So if you see the products, so in case of so row independent termination, so termination signal is a stage of 30 to 40 nucleotides on RNA transcript consisting of many GC followed by series of U. So they will form a stem loop structure. So you will see, so they will form a hairline structure or a stem loop structure, and finally to terminate the transcription factors. So this is the just example. You will see here. This is the one protein. So this is the from DNA to mRNA is taking place, and you can see here. So how it is going to the loop, a stem loop formation, and when a stem loop formation will take place, your transcription is going to be terminated. Okay. So that is called stem loop structure alters the conformation of RNA polymerase, leading to pause of RNA polymerase moving. So when a stem loop formation forms, so it will change the conformation. So means your whatever RNA polymerase is just moving, so it will just stop due to the conformation change. Then the competition of RNA-RNA hybrid and DNA-DNA hybrid reduces the DNA-RNA hybrid stability. So that is again an important factor and causes the transcription complex dissociated. And all the base pairing more unstable is RU and DA. So all this is all about transcription in case of prokaryotes. Thank you.